Um, it's such a pleasure and honor to welcome to Living Histories Professor Alice Pyne. Please take it away, Alice.
Thank you so much for the invitation. I feel really honored to be here. And when I was putting this together, I looked for lots of photos of my early years and realized that they're all in boxes at my parents' house. And so as a result, I've decided to talk through my journey to where I've become today without slides. So hopefully this brave experiment pays off. So I was born in London in the UK, but when I was about 4, my parents moved to the countryside, thinking that a young girl should grow up playing outdoors rather than in the city. And I grew up as an active kid. I was climbing trees. I was playing in the garden. I was musical, and my parents really supported me in doing lots of different things. And I never, in my wildest dreams, would have expected to end up a scientist, let alone a professor at a university. But one thing that I think when I've been reflecting on this that I always enjoyed was finding out how things worked, whether that was taking apart a flower that I deadheaded in the garden, or rewiring a plug with my dad, or learning to build a speaker from scratch. It was always understanding things, how things work, that motivated me. And I think that's something that really has maintained a key point through my career to date. As I grew up a little more, I went to school to do my GCSEs and A levels. And again, I enjoyed most subjects. I call myself a sort of interest magpie, and I enjoyed science, I enjoyed music, I enjoyed languages. And I chose a really wide range of things, narrowing that down to Latin, maths, Spanish, and physics, a very heterogeneous bunch of subjects for my final A levels. And I was interested in all of them. And I think the thing that linked what my interests in those were today is actually, I think I've always been really interested in seeing patterns in things, whether that's in musical scales or musical chords, or whether that's in equations of motion, or whether that's in languages and how they're designed. And I actually applied to university for Latin. So I wouldn't call myself your standard sort of physicist in training. And it was during my A levels that I realized I wanted to do a subject where I could find out how things worked and gain concrete answers. And I always laugh a little when I say I then went on to do physics, because I think anyone who does an undergraduate realizes you have to learn the same thing a few times to really start to even fully understand it or be able to explain it. But I chose physics, and I went off to university for 4 years for my integrated master's course. And again, I enjoyed a huge range of subjects from particle physics through to mechanics. But I was always happy in the lab. I think that's one of the unifying things. Again, whether that was building a radio, which was one of our labs, or whether that was doing optics and waves and designing new optics experiments. I was always happy in the lab tinkering around with things. And it was really my final year project where I think I really started to click with the subject I was learning, and I really started to understand why I was doing these courses and what they would help me to understand more. So I chose my final year project, and I didn't get my first choice, I didn't get my second choice, but I got my third choice. And that was designing a high-speed atomic force microscope with Professor Mervyn Knowles at Bristol University. And I was extremely excited to do it. And I think one of the things that I really enjoyed was in my undergraduate, we did projects as pairs. So I got to work with a lab partner. And I think for a project that ended up having to take place largely at night due to building works that were happening directly next to the microscope I was using, and also on samples and experimental setups that we had to design from scratch, I don't think I could have done it without having someone to bounce my ideas off. And I think that's still how I work today. I work much better when I've got collaborators and colleagues who I can go and chat to about ideas, and together we can work out the best way of interpreting whatever we're looking at. So I really, really loved working with Will. And we actually looked at dental enamel and how it dissolved in citric acid in the presence and absence of fluoride. And I think, again, it was doing a project that had such a tactile link to your everyday, that you were able to understand a little bit more about what your everyday life was like. You know, don't drink fizzy drinks, they dissolve your teeth. We could watch that happen in real time. And I was offered a PhD within the group to stay on. But I'm actually the first person in my family to have a degree. and I wasn't sure I wanted to commit to a degree, to a PhD at all. So I went to work in a couple of research labs over the next year, working in clean rooms, doing silicon wafer manufacturing, which is quite far from where I am now. I was very proud of my 50 nanometer resist, which I think ages me quite a lot. And I also worked on environmental ecotoxicity of nanoparticles. And I realized I did actually really love microscopes, and I wanted to do a PhD in microscopes. And I applied to quite a few labs, and I ended up taking a PhD in London with Professor Bart Hoogenboom, designing and building atomic force microscopes in a biophysics lab, so to probe a range of samples. And in my PhD, I looked at lots of different things. I spent a lot of time looking at antimicrobial peptides and working with chemists and biochemists in the lab and learning a lot of new things like peptide synthesis and how to make cationic lipid bilayers and negatively charged lipid bilayers. But the thing I loved was actually sort of a chance encounter. We decided to start a collaboration with BRCA, and I met them at a conference, and we got chatting about things, and they invited me over to their headquarters in California. And I ended up actually spending a lot of my PhD traveling between London and California, designing experiments and working in the lab in their headquarters and testing out new types of manufacturing to try and improve the resolution of our microscopes. And it was actually there that I had one of the real breakthrough moments of my PhD for me, which was seeing the double helix of DNA on a single molecule. And I'll never forget it. It was 10 o'clock at night because experiments work better at night. And it was with my colleague Andrea Slade from BRCA. And we've been trying to do this for 2 weeks, and it was just before I was leaving, and it was when they were threatening to turn the power off for the whole building for a reset. And we finally got these experiments to work. And it's one of my papers I'm still very proud of today, but it also really kick-started my career in this area. So sort of finishing my PhD, I managed to develop my love of microscopes even further, and I realized that it was DNA that I was really interested in. I was really interested in how its mechanical properties inferred its function as well as its sequence. And that was due to the results I'd had, but also the meetings I'd had with Professor Sarah Harris, who's now a colleague of mine and has become a very good friend, and Professor Tony Maxwell, who was one of the key people sort of guiding me through the early parts of my career. And one of the things they said was, you know, you've got a new technique, you've developed this technique, try and apply it to things. What could you apply it to? And so during my postdoc and my first postdoc fellowship, I spent a lot of time working in an area that I'd never really come across, which was DNA topology. And I went to conferences where I learned about things like ataxia, and where I saw mouse models, and where I saw, you know, clinical mice work and clinical trials, as well as seeing things that I was much more comfortable with, like X-ray crystallography and cryo-EM. And that started me and my group, which started in London, on our kind of journey of trying to develop new tools to understand the structure and topology of DNA and how that influences biological processes. So it was in 2019 that I was looking for a permanent faculty position and realized that probably wasn't going to be in London for me. And I decided to make a big move back to the countryside, but a bit further north to Sheffield, just on the edge of the Peak District in the UK, and to start my group again there. And it's been a brilliant choice. Moving just before the pandemic didn't have its, wasn't straightforward, and setting up a lab during the pandemic wasn't straightforward either. But I'm now incredibly lucky to work with fabulous colleagues. There's a lot of interest in what we do here, but also to have collaborators around the world who want to work with us to help us develop new ways of seeing DNA structure and understanding what DNA might be doing within a cell. And I think a really big part of that was actually learning to do computational work and diving into computational image analysis, something I had basically no formal training in. My undergraduate programming was all done in Visual Basic, which I would not recommend for doing anything. And so that's become a really key focus of our group, is bringing together experiments and new microscopy methods with quantitative analysis to try and understand the structure of DNA. So yeah, sorry, it's less clear without.
Is that
Yeah, I'll finish there.
okay?
The timer stopped working, so I'm guessing I was about right.
Yes, sorry. You're in good times, Alice, and we have time for a couple of questions. Let me first thank you on behalf of the audience and clap on behalf of the audience. Audience, you're welcome to send me questions via chat. Let me get started by asking you, To first confirm that you said you're the first person in your family to have a degree. Is that what you said?
Yeah, that's what I said.
Um, is that, in your view, somehow tied to how intrepid an explorer you've been through your trajectory? Because,
Maybe because I think I didn't know how I should take that trajectory. I think I had no idea what an academic career was, um, or what it might look like. And I think I've been really, really lucky to be encouraged by my parents to do whatever I wanted to do, whether that was, I think they thought I'd be a musician, not a physicist. But I think I was really lucky to be guided in that way. But I think also I've been really lucky to find a lot of collaborators who do have experience and many degrees who've helped inform me of, the paths I should be going down. And I've still chosen my own. I think I'm probably very stubborn. But I think it's been wonderful to have such a range of brilliant collaborators who've always taken interest in me. But yeah, I think it definitely, I hadn't really considered it as not being normal to be the first person in your family to have a degree. But I think as I progressed through my academic career, I'm obviously very privileged in other ways, but I think it's become more obvious to me that maybe I've followed a different path, a more circuitous path, which I think we've heard from other people in living histories is a normal way of getting there. But I think that was partly due to a lack of understanding of what that path should look like.
that leads nicely to the next question I want to ask you, which is, you called yourself a pattern spotter,
Mm-hmm.
and your interests also reflect that in both music and physics. Would you please tell us if there's a hidden pattern in all the meanderings you've done to get to this part?
Oh, yes, I think there are. That's a really lovely question. Um, I think the patterns are incredible, supportive people. Um, I could name Sarah Harris as one of the people that's been supportive of my career, and I'll name her many times. But I think, one of the things, I could go back to my lab mate, Will Marks, from the first time I fell in love with microscopes, and he was there in the evenings with me working on the microscope. I think it's supportive people, and that can be, I think we often talk about mentors, but maybe less often we remember to reference the people around us, supportive lab mates, supportive colleagues, even if they don't work in the same area, people that say, oh, have you thought about that? And also, I think getting comfortable not knowing anything. So going to a conference where people were talking about ataxia in mice, when I was a physicist who built microscopes, and saying to people, asking the questions that felt silly. And I think that's enabled me to grow a lot in my career. So yeah, I think having supportive people at all levels below me, my group were incredibly supportive. Alongside me, I've always had really supportive colleagues, and above me, amazing supportive mentors, but also, that willingness to be the person to ask the silly question because they don't know what they're getting themselves into.
Wow, thank you so much. On that super inspiring note, I am thanking you again in closing.